Hi everyone, this is Anurag again. So I know I said that my content will be mostly heme path heavy, but this month I was on molecular pathology rotation and I came across next generation sequencing. And it is a truly fascinating subject. I mean, this was one of the things when I was reading about it. At most of the steps of this process, I was going like, wow, that's brilliant. It's genius. I mean, how can people, how can somebody even think about this process? I mean, I'm stupid enough and I'm just thinking about what the next episode of House of the Dragons would do. And people have just thought about this miraculous thing here. So I thought, why not just make a video about next generation sequencing, just the very basics about what happens in next generation sequencing and just the basic process. So I'm not going to go into too detail, but just the basic process of next generation sequencing. So there are a few prerequisites before we go into this lecture. That is, you should know what is DNA replication and what is the polymerase chain reaction. So I will not go into details of those. So now let's head into into the next generation sequencing. So first of all, we have the sample. So to do the sequencing, we first need the nucleic acid from the sample, and the nucleic acid is inside the nucleus, which is inside the cell, which has a cell membrane. So we have to remove the other stuff, that is the cell membrane, the proteins and everything, and isolate the nucleic acids first. So this is accomplished by means of first cell lysis. That is what happens in cell lysis is that we use various methods including detergents or solvent, heat, osmotic gradient or sonication to damage the cell membrane and remove the lipids from the solution. So once the lipids are removed, now we have the proteins that we need to digest. So proteins are digested with the help of an enzyme called proteinase K at 56 degrees Celsius. So now we have gotten rid of the lipids, we have gotten rid of the proteins. We have the nucleic acids, but now we need to extract and purify the nucleic acids. And this is done with the help of different techniques like phenol extraction or ethanol salt precipitation, or there's a third technique called chaotropic salt slash silica column slash silica bead extraction. So each of these methods, if I go into detail, it will become a very long process, and since the main purpose of our video is to focus on the process of NGS, so I'm just gonna mention these things here. But these methods mostly utilize the differential solubility of nucleic acids in various solvents and help in purification and extraction of the nucleic acids. So now once we have extracted the nucleic acids, we need to determine whether the nucleic acids are in sufficient amount, that is the quantity, as well as the quality of the nucleic acids that we have extracted, so that we need to know whether they are good enough to perform a sequencing evaluation or not. And again, there are different methods to do that. So one is a spectrophotometric absorbance methods. They can be fluorescent stains, and they can be probes that can be used to determine the quantity and quality of the nucleic acid. So after we have done all this, that is, we have extracted the DNA, we have extracted the nucleic acid, we have ensured that it is of a good quantity and quality. Now we move on to the next generation sequencing. So it can basically be divided into 5 steps. That is, first, that is isolate the genomic DNA. That is what we did before. Then create a library, and I'll explain what this means later on. Then we have to separate the fragments and create clusters. Again, these are just short headings, and I'll go into details of each of these steps later on. And then we have to sequence the clusters, and then we have to analyze the data. So we have isolated the genomic DNA, and this was what we had discussed so far. That is, first we lyse the cells, we lyse the lipids, we lyse the proteins, and then we extract the nucleic acid. So now, first of all, the genomic DNA that we have isolated, we now need to break the DNA up. We need to break them up into different fragments because that will help us in sequencing. So there are different methods for breaking it. There can be ultrasonic disruption, and there can be tagmentation, and these are really fascinating techniques. Let's first go into ultrasonic disruption. So we just have a container, we have the nucleic acids, and then we just buzz it with sound. And these are like bzzz, and these just go in, and then they just break the DNA into small fragments. So the problem with ultrasonic disruption here is that, as you see, that the fragments have different sequences at their end. So it's just like sound waves that is kind of like, going in and, creating bubbles in the solution. And these bubbles break the phosphodiester bonds between the nucleic acid and break the DNA into different fragments. So each of the fragments would have a different sequence at their ends. And also, if you see that the fragment ends are also uneven, if you see here that this end is slightly longer than this end, and similarly this end is shorter than this end, so these are uneven. So that is why we need adapters. So what are these adapters? These adapters are truly fascinating stuff and fascinating sequences of DNA. These are actually chemically synthesized double stranded DNA that have specific sequences. These adapters have these adapters attached to these ends of the DNA, as we had talked about, and they help in multiple things. So one side of the adapter has a surface, has a sequence that is complementary to the solid surface. What it means is that, as we'll see in the later steps of the next generation sequencing, we have to immobilize the DNA fragments on a solid surface, and there are certain sequences that are already attached on the solid surface which help us stick these fragments to those sequences on the solid surface. So this adapter sequence actually helps stick the fragments of DNA on the solid surface. Then there is a sequence primer binding site. Sequence primer binding site is the site where actually the DNA primer and the DNA polymerase will bind and will help in the actual sequencing step. And we'll go into the details later on as we go through this process of the next generation sequencing. Now, that is the barcode sequence. This is a truly fascinating thing that, I mean, it was like, for me, when I read about it, it's like genius. So, I mean, suppose you're doing a next generation sequencing process. So what if you had so many samples that you could not run one sample per test because these things take time? So what if you wanted to run all the samples together or multiple samples together in one process? So this barcode sequence actually helps in identifying different samples within the same test. So you can actually mix different samples together, but after attaching these adapters, you can only mix them after attaching the adapters, and each of these adapters will have a specific sequence for a particular sample, so that later on the computer or the software can decode what fragment or what DNA belongs to what particular sample. So just to explain it again, suppose we have multiple patients, like patients A, B, C, D, and we mix the samples of all these patients together. So each of these patients will have a specific barcode sequence that is attached to the adapters of their fragments. So the software will be able to detect that specific barcode sequence and will be able to tell whether that particular fragment belonged to patient A, B, C, or D. Truly fascinating, right? So, and then the other, so this was ultrasonic disruption. That is, we use sound waves to disrupt the DNA into multiple fragments, and then we add adapters. Another method is tagmentation. This again is a very fascinating technique. So what it utilizes the transposase enzyme made by the E. coli transposon. So if we just jog back our memory to medical school, what were transposons? Transposons were actually mobile genetic elements. That is, they were like a sequence of nucleic acids that just moved from one place to other. So they used the enzyme called the transposase. What does transposase do? Transposase attaches to the transposon, that is, it carries that particular mobile sequence with it. It goes to a particular point in the DNA, cuts the DNA, removes a part of that DNA and inserts the transposon into that DNA. So what we do is that we, instead of adding the transposon to the transposase, we attach the adapters to the transposase enzyme, and this is shown here in the figure. So this red DNA is our DNA of interest that we want to do sequencing on. This yellow diamond, it is the transposase enzyme, and this blue is the adapter. So we have attached the adapters to the transposase enzyme. So what this transposase or this yellow diamond would do, it would make cuts here. So let me just use another pen just to highlight it better. So it would make cuts in the DNA here and would attach these adapters at these cut sites. So it would make cuts here and attach these adapters to these cut sites. So ultimately, after both of these steps, that is ultrasonic disruption and adapter addition, as well as this fragmentation, what we get is our fragment of DNA, which is attached with adapters on both the ends, that is the 5' end as well as the 3' end. So now we have gotten the fragments that have the adapters attached to them. So the next step will be to separate out these fragments so that we can sequence each of these fragments individually, and we'll go through the process now that how to separate out these fragments. So these next generation sequencing machines usually have, and this process, I mean, so the ones that we usually use or what I have heard of is from Illumina, and Illumina uses something called sequencing by synthesis. So they have, their machines usually have something called a flow cell. So what is a flow cell? So flow cell is like a, it's like a slide, and it has multiple channels which are highlighted by blue here, and the white is actually the flow cell and blue are the multiple channels inside it. And so we pour the sample into these channels. So let's just take a magnified view of one of the parts of these channels here. So the blue represents the surface of the channel. So on the surface of the channel we have these are actually nucleotide or oligonucleotide sequence. So oligo basically means few. So there are few nucleotides that are attached to each other. And remember that we had talked about the adapters before, and they had a sequence that is complementary to the solid surface, that is this area here. So this sequence is actually complementary to these oligonucleotide sequences here. So this helps in the attachment of the fragment to this solid phase. So as the fragment flows and under appropriate temperature condition, so we see here that this fragment gets attached to one of these oligonucleotide sequences here, and this is how we separate out the fragments. So now what happens is that you see that the fragment that we had created had adapters on both ends. So there was an adapter here and there is an adapter here as well. So this adapter is also complementary to one of the so this adapter here is also complementary to one of the oligonucleotide sequences here. So this will bend over and create a bridge, and this is called a U-shaped bridge, and that so this is what's called a bridge in next generation sequencing. So now we have to do a PCR of this bridge, because we just have, say, one fragment of that particular DNA. We want to amplify the number of fragments so that we can sequence them properly. So there is this PCR primer which attaches to one of the ends and causes multiplication, and so that, as you see, that there is this replication of the strand that occurs with the help of PCR, and then repeated PCRs creates clusters of fragment, and each cluster of the fragment consists of multiple copies of the same fragment. So let me just explain what this means. I mean, I know it's difficult to, I mean, it was very difficult for me to understand what this actually means is. So suppose we are looking at it from above. So this is so suppose I'm drawing a flow cell here. And this entire thing represents, say, this cluster. So this round shape has this multiple DNA fragments here. Similar to these, there will be multiple clusters on this flow cell. Now each of these clusters will have multiple fragments of DNA, and each of these fragments in one cluster would have the same sequence. That is, all fragments in this cluster would have one sequence. All fragments in this cluster would have one sequence. All fragments in this cluster would have one sequence here. Because why do they have one sequence? Because there was initially just one fragment that attached, and then it underwent multiple cycles of polymerase chain reaction or PCR, and so there were multiple fragments with the same sequence here which are in one cluster. So understood that each of these clusters would have multiple fragments of the same sequence. So one cluster will have one sequence. So now we have generated the clusters, and now it's the time for actual sequencing. And the actual sequencing actually includes called the sequencing primer, which I am representing here with a white rectangle. The DNA polymerase I am representing with a yellow sphere, and then there are fluorescently labeled nucleotides. That is, each of the nucleotides would have a specific color. I've just arbitrarily given colors, like white is adenine, then orange is G, then green is T, and then purple is C, and it's not these colors. I just arbitrarily given them. And then the fluorophore, that is the thing that's giving the color to the nucleotide, it acts as a blocking agent. This is very important, and I'll explain why this is very important. So this fluorophore acts as a blocking agent. That is, it does not let the DNA polymerase proceed further or add more nucleotides until the fluorophore has been removed. And this is a very important part in determining sequencing, in determining the sequence of the particular fragment. So what happens here is, so we have again our fragment here. We have the solid phase here where the oligonucleotides are attached, and our fragment is attached with the help of the adapter to one of the oligonucleotides. So here we have the sequencing primer, we have the DNA polymerase. Let's say, for example, the DNA polymerase adds the first nucleotide, and just for example, let's just say it's T, and we remember that that T had a green signal. So now the T has been added to the sequence here. So the fluorophore, or that is the thing that's giving the color to our nucleotide, that is nucleotide T, it blocks this DNA polymerase. Oh, I just accidentally erased everything. Well, thank God for the undo button, right? So the fluorophore on T will block this DNA polymerase and will not let the DNA polymerase move further. So now we only have the green signal. So if our fragment had A and the DNA polymerase had added T to it, there will be a green signal. And this green signal is read by the software or the computer, and the computer also reads the signal that's generated on every cluster. Now suppose there was a fragment where there was no T that was added, so there will be no signal from there, and that is also read by the computer. Now with the help of various steps like washing and everything, the fluorophore is removed, and now the DNA polymerase is free. Now actually I can use it. Oh no, I cannot use the erase button. Sorry. So now after washing, the fluorophore is removed and the DNA polymerase is now freed from the influence of the fluorophore, and it can move further and add more nucleotides. And so the entire process is repeated. This entire process is repeated in sequence using different nucleotides. Say for example, we use T here, now next we use G, and then again the computer will read the signal and the fluorophore will block the DNA polymerase, and once the software has read all the clusters, the fluorophore will be removed, and again we use now another nucleotide, say A or T or some other or any other depending on the nucleotide that we want. And this is how the sequencing process or the reading process goes further. And there are different primers that will actually detect the barcode sequence. You remember the barcode sequence that assigns, that is specific for a particular sample and helps us to sequence multiple samples together. So there are different primers that will read a specific barcode sequence and will let the software know that what clusters belong to what sample. So now that we have sequenced, now it's the time to analyze the data. So the barcode reads, so the adapter barcode reads, they are used to sort the cluster into different samples, which we just talked about, that there are specific primers that will read the barcodes and will assort the different clusters to different samples or different patient samples. So now the second thing that we do is we use a reference genome, and the reference human genome is available from the Genome Reference Consortium website. And now each of the reads, that is each of the fragments that we have read, is aligned to the reference genome to get the sequence and see if there are any differences. So what do we mean by this? So say I would show this with the help of a figure. So the green here is the reference genome. So this is just an arbitrary sequence. I just made whatever sequence I wanted using A, G, T, and C. And the reds represent the reads. So as you see, there are one area of the reference genome, say for example here, is read multiple times with different reads, because the fragments can be of different sizes and lengths, and so therefore we can get different reads. And all of these reads which are represented by red, they are aligned in respect to the reference genome, and then we determine the sequence. And another term that we usually use with next generation sequencing is called the read depth. What does that mean? So read depth usually means the number of reads that cover a location, that is called as a read depth. For example, if we are talking about this location here, that is the A location here. right in the center and which highlighted by the blue box. So the number of time or the number of reads that cover this location is called the read depth. So here we see we have read number 1, 2, 3, 4. So there are 4 reads here that are covering this location as compared to say location T here. So location T here, we only have 1 or 2 reads that are covering this location. So we are much more confident in saying that this location A is actually an A, whereas this location T could be something else as well. So this read depth helps us develop confidence in the sequence that we have determined. And usually a depth of 20 to 30x is pretty good. I mean, I only showed 4 here because of, I mean, I didn't want to cover the entire canvas with the number of reads. And so this is what is called as the read depth. So this was in brief the next generation sequencing, and my reference was the Molecular Biology 3rd edition book. And just to go over in summary, it includes first isolation of the genomic DNA, which we did. Then we break down the DNA using ultrasonic disruption and tagmentation. We attach the adapters. Then after attaching the adapters, we now have to separate the fragments, which is done by putting it on the flow cell. Then we separate the fragments. The fragments form a U-shaped bridge. These bridge undergo polymerase chain reaction, so which helps us to form multiple clusters of particular fragments. And then we undergo sequencing using the sequencing primer, the DNA polymerase, and the fluorescently labeled nucleotides. And then after which the reading is done, we align it with the reference genome and determine the sequence and determine if there are any alterations in the DNA that we are sequencing in respect to the reference genome. So this is just a very brief video and I hope you liked it. Please let me know if I need to make any changes, or if you would like any other particular topic that I need to talk about, that you would like me to talk about. And thank you so much for listening.